Chapter 8, How Cells Release Energy. In the previous chapter, we went over how organisms that can do photosynthesis take light energy and they convert that into glucose. Specifically, what did we say? We said that light energy will excite electrons. The source of those electrons is the splitting of water. As water splits, it releases oxygen that releases a byproduct. There are a series of steps, and the light-dependent reaction, that was the first one, we converted that light energy, that hydrogen, those electrons, and we made ATP and NADPH in the light reaction. Those are energy carriers, short-term energy carriers. We also did what? We made oxygen as a byproduct. The next step, the light-independent reaction, that Calvin cycle, that's going to take those products and cycle them around and make what? Glucose. What goes in the Calvin cycle? Carbon dioxide. That's going to be the carbon dioxide, the carbon dioxide will be the source of carbon and oxygen in glucose. Okay, so what we're doing here is that in the light-dependent reaction, that ATP, that's like the $20 bill. Light-dependent reaction, we're making those $20 bills into one big $1,000 bill. That's the glucose. This chapter is going to take that $1,000 bill, that glucose, and break it down into a bunch of $20 bills, which is ATP. Okay, so we're going to kind of do the opposite. Okay. We're good here. So this is what we're doing. We're going to take glucose. See that picture here? We're going to take glucose and oxygen, and we're going to do is a series of steps, and we're going to release carbon dioxide, ATP, and water. Okay, so we're going to make that $1,000 bill, which is glucose, into a bunch of 20s, which is that ATP. So let's start at the top. It says what? Carbohydrates break down, start in the cytoplasm with glycolysis. Okay, so the point here is what? Glucose is going to enter, and it can be other molecules too. It can be lipids, but we're just going to say glucose for now, and then talk about some other things later. But let's just say glucose for now. That's going to start in the cytoplasm, and that's going to enter glycolysis. That glucose is going to break down in a process called glycolysis. Now, the products of glycolysis can go either into the pathway to the left, which is fermentation, or the pathway to the right, which is aerobic respiration. Okay, so this slide is just saying that two options for the products of glycolysis is aerobic respiration inside the mitochondria in eukaryotic organisms, or fermentation, which is that left pathway. So, but you should know what these two are. Okay. As we said, after glycolysis, that's step number A in this picture, the pathways can diverge, and either that the products of glycolysis go to fermentation or to oxidative metabolism, or here aerobic respiration. Okay. Also look at the number of ATPs being made. Fermentation makes less ATPs. Aerobic respiration makes more ATPs. So first we're going to look at aerobic respiration, and we're going to look at glycolysis first. Okay, but what comes out of glycolysis does not necessarily have to go to aerobic respiration. So let's look what goes in. Glucose goes in, oxygen goes in, carbon dioxide come out, and water come out. You should know what goes in and what comes out. Okay, so what we're going to do is look at stage 1, which is glycolysis, then stage 2 and 3. Okay, then we're going to go over each one a little bit more in detail. So first stage is glycolysis. What's going to happen here is glucose comes in, 2 NADH come out, and 2 pyruvate come out. The pyruvate are a 3-carbon compound. Look at ATP here. What's happening? 2 ATP go in, 4 ATP come out. So I'm spending 2 ATP. I'm making 4 ATP. So what's my net increase in ATP? Well, that's 2, right? I spent 2. I made 4. After I take care of my debt, I've made a net of 2 ATP. So take home, glycolysis makes a net of 2 ATP. Glycolysis makes a net of 2 ATP. You need to know this. Also, you need to know that glycolysis makes 2 pyruvate and 2 NADHs. So glycolysis makes 2 NADH. Glycolysis makes 2 pyruvates. Here's a summary. You should know this. Now we're going to look at step 2 and 3. So step 2 is the Krebs cycle. Krebs cycle, this is going to take the products of glycolysis and it's going to put them in a reaction that will convert them into some other energy carriers. We're going to look at the Krebs cycle in detail later. But what do you know about the Krebs cycle? Pyruvate comes into the Krebs cycle. CO2 comes out of the Krebs cycle. Some ATPs come out of the Krebs cycle. And more importantly, NADH and FADH2 come out of the Krebs cycle. And we can convert that NADH and FADH into ATP during electron transfer phosphorylation. Okay, so what comes in, what comes out. Also, in the NADH, it's worth 3 ATP. FADH2 is worth 2 ATP. Okay. Final stage, step 3, is the electron transport chain, or electron transfer phosphorylation. This is going to take products of the Krebs cycle, and it's going to make those products, the NADH and FADH2, into ATP. So that NADH and FADH2 are holders, or they're proxies. They are holders for ATP. Okay, so we can take those and exchange those and make ATP. We're going to talk about how that's done in detail later on. Okay, so what we're going to do is, I just talked about 3 steps. Glycolysis, the Krebs cycle, and then that electron transport train, electron phosphorylation. So what I'm going to do now is talk about each one of those steps in much more detail. And I'm going to start with glycolysis. Okay, so the previous material was just an overview, a review of what's going on. This is the specific parts you need to work on. So what's glycolysis? Well, let's just say what goes into glycolysis. The important point here is what goes into glycolysis. Glucose goes into glycolysis. NAD goes into glycolysis, and ADP goes into glycolysis. What comes out? Pyruvate, ATP, and NADH. This is glycolysis. These are the steps of glycolysis. I want to show you the important steps. What you see on the right is the entire step, all the steps together. We're going to look at each one of these steps separately, and I want to tell you the important points. Okay? So the first part is glucose. It starts at the very top. See the 6 black balls? That's 6 carbons. ATP is going to add a phosphate, that's phosphorylation, 2 times. At the end, it's going to make something very unstable that will break apart into 2 3-carbon compounds. That's what you need to know. You don't need to worry about the names like glucose 6-phosphate. You don't need to worry about fructose 1,6-bisphosphate. No, no, no. Just understand that we start with a 6-carbon compound and 2 ATPs are added to it. That 6-carbon compound at the bottom will break apart into 2 separate molecules. Okay, now we're going to look at, do you see at the very top it says 2-p-gal? Okay, don't worry about what p-gal is. Just know that those are 2 3-carbon compounds. 2 3-carbon compounds. And we're going to do some enzymatic steps here. We're going to do some react stuff to them, right? And what happens when we do all these things to it is that ATP is being released and NADH is being formed. Okay, so as we change this around, we're going to form ATP as a result. So what do you need to take home from this? 1, 2, 3 carbon compounds will have ATP. Well, let me say it this way. The 2, 3 carbon compounds at the top will have some enzymatic reactions done to it. Some enzymes are going to work on this. End of the day, they're going to make 4 ATP from 1 glucose. So see the 4 ATP on the right? 4 ATP come from 1 glucose. Also, it's going to make NADH. At the end, you need to know this. It makes 2 pyruvate molecules. Pyruvate has 3 carbons, and it's going to make 2 pyruvate molecules. Okay, so what goes in, what comes out of glycolysis? Let me go back to that. What goes into glycolysis? Glucose and 2 ATPs. What comes out of glycolysis? 2 pyruvate at the very bottom of the blue chart and 4 ATP. Because you use 2 ATP and you've got 4 ATP, we say there's a net release of 2 ATPs from this reaction. Okay, so now what we're going to do, we're going to go to step 2. We said before there's 3 steps: glycolysis, the Krebs cycle, and oxidative phosphorylation. We're going to look at stage 2 now. This is the Krebs cycle. What's going to go into the Krebs cycle? Where does it occur? Important point here is what? This occurs in the mitochondria. This occurs in the mitochondria. Also, this requires oxygen. This requires oxygen. You see aerobic respiration? That aerobic tells you, like aerobics, you're dancing around, all that, this requires oxygen. So that pyruvate is broken down, and look what happens when pyruvate's changed. Pyruvate goes from 3 carbons to 2 carbons. Look what happens. A CO2 comes off. You know when people say we breathe out carbon dioxide? Well, this is why we breathe out carbon dioxide, because as we convert this pyruvate, which was started out as glucose, and we remove that carbon dioxide, or remove carbons from it, that carbon comes off as carbon dioxide. Okay, so that's why we release carbon dioxide. Also, look at what's happening. We're releasing carbon dioxide from the Krebs cycle. So what happens with the Krebs cycle? First of all, let's talk about the Krebs cycle. The Krebs cycle happens in the mitochondria. It's going to release CO2, and it's going to release a few ATPs, and it's going to release the NADHs and FADH2s. These NADHs and FADH2s can be exchanged for ATP later on. Okay. So just look at the cycle here. What's happening? We have a stepwise removal of carbon. Let's look at that citrate. See that citrate is about the 10 o'clock position. If you thought the Krebs cycle was a clock, xylotate's at about 3 o'clock, well, about 2 o'clock. Citrate's at about 11 o'clock. Let's start that citrate. Look at it going counterclockwise. A carbon, 2 oxygens come off. That's the CO2, carbon dioxide. NADH is made. Another CO2, carbon dioxide, comes off. NADH is made. ATP is made. FADH2 is made. NADH2 is made. What do you need to know about this? These are all energy sources. ATP is an energy source. The NADH and that FADH, notice how it looks like squares? It looks like kind of like a check. Well, they're kind of like a check because we can take those to the next step and cash that check in for real ATPs. So think of these as checks for $50 bills. Okay, we can take these in and we can change them in on the next cycle and make ATPs. So these are almost as good as ATP. Okay, so let's make some ATPs. But the point is this: the Krebs cycle makes a lot of energy. Well, I should say it this way: it releases energy. It releases a lot of energy. It's not making energy, but it's releasing energy. Okay, the Krebs cycle is releasing a lot of energy. It's releasing it from potential energy in that form of the citrate to another form of potential energy, which is that ATP. Using another example, I could say that I put the pyruvate's like a $500 bill. And this cycle is going to release a couple 20s, and it's also going to release a couple checks that, that says, take it to the next step, I'll give you $50 there. Okay. Okay, so now we're going to talk about what we do, that third step. We said there are 3 steps we're going to cover, right? This is the third step. Phosphorylation. Electron transfer phosphorylation, or oxidative phosphorylation. Happens in the mitochondria also. We're going to make a gradient of H+, and let me show you a picture of that in a minute. But by making that gradient, we're going to make ATP. So what happens? That NADH, FADH, too, that is going to be a source of electrons. Those electrons are going to go through something called an electron transport chain or electron transfer chain shown in this diagram. That's going to make a lot of H+ protons in the center. You see all the H+ in the outer mitochondrial compartment? The actions of NADH and FADH2 make those happen. They make that concentration gradient form. Oxygen, see why we need oxygen? After all those electrons have done their job in making that H+ gradient, we've got to get rid of those electrons. Also got rid of, and so we take those electrons and the protons and combine them with oxygen, and that makes water. So that's why you need oxygen. You have to get rid of these products. How you get rid of them is you combine them with oxygen. We take these protons and we run it through. Now look at the far right. See that blob? It looks like a marshmallow going through the membrane. Well, that H+ is going to go through that, and it's going to physically spin. And as it physically spins, it's going to make ATP. So this thing spinning will make ATP. It's going to make a lot of ATP. But the greater point is what? NADH and FADH, by their actions, they make, and that's their actions on the far left, are going to make that structure on the far right work to make ATP. So NADH and FADH2 are converted in a sense, or their actions make the release of ATP on the far right. Okay, and it's going to make a lot of ATP. Typically, glycolysis is going to make 2 ATPs. The Krebs cycle is going to make 2 ATPs, and the energy carriers that we talked about, FADH and NADH2. And then the last step is going to take those energy carriers and convert those into a lot of ATPs, 32. And again, let's not lose sight of the point here also is what? The energy, all of this energy ultimately comes from the sun. Okay? The NADH, that FADH2, I want to make this point, and this is an important point. Let me go back to the start here. Bear with me. Okay. Stand by, stand by. Okay. Let's look at this picture. You see glucose at the very top? The energy that made glucose is from the sun. Okay? That glucose at the very top going to glycolysis, the energy for that was from the sun. Now, in a plant, it made ATP and went to glucose. In us, we take that glucose and we go back to ATP. Okay, that's kind of the point. And that ATP energy that we use is from glucose and some other things, lipids, some other molecules. But ultimately, we trace that back. The energy that's in there, or the chemical potential energy, comes from the sun. Okay, that's the greater point here. Okay,